Welcome to Học Tiếng Anh với Joseph, where we build vocabulary and practice pronunciation through reading. Continuing with The Cat in the Box, we will read story number 55, and it's called The Birth of Biochemistry. Even at the end of the 19th century, there were still respectable scientists, including Louis Pasteur, who argued that there was something special about the chemistry of life and that some life force was involved in what were called vitalistic processes. The final refutation of this idea came in 1897 from the work of the German Eduard Buchner. Buchner decided to tackle the question of fermentation, which divided opinion at the time. Fermentation is a process that takes place without using oxygen, in which living cells convert foods such as sugar into simpler compounds such as alcohol and carbon dioxide. And the energy that powers the cells is released. Fermentation is a common process in living things, but Buchner studied the simple process of alcohol production, which involves yeast. Fermentation was seen by many to be a physiological act inseparably linked with the life processes of yeast. Although Friedrich Wöhler had poked fun at the vitalism idea as far back as 1839 by satirically summarizing the argument as, in a word, these infusoria gobble sugar and discharge ethyl alcohol from the intestine and carbon dioxide from the urinary organs. Yeast is indeed a living organism, and yeast was essential for the process. But was this because the yeast cells were alive, or because they contained some chemical substance that encouraged, catalyzed the conversion of sugar into alcohol and carbon dioxide without any need for vitalism? Could fermentation occur without the presence of living yeast cells? The only way to find out was by experiment. Buchner had long been interested in the problem, and when he was appointed as professor extraordinary for analytical and pharmaceutical chemistry at the University of Tübingen in 1896, he was able to carry out the work on a large scale in a fully equipped laboratory. By January 9, 1897, he was ready to send his key scientific paper on alcoholic fermentation without yeast cells to the editors of the journal Bericht der Deutschen Chemischen Gesellschaft. Yeast cells are like small bubbles filled with a semi-liquid substance, the protoplasm, surrounded by a comparatively firm cell membrane. To investigate the chemical composition of the cell contents, the experimenters had to remove the membrane by crushing it, because if they used any chemically active solvents or high temperatures, this would alter the chemistry they wanted to study. And it was important that the process should be completed as quickly as possible to minimize the possibility of any change while the experiment was going on. So Buchner started with living yeast cells, but then treated them to a series of processes that killed them and reduced them to their constituent chemical parts by purely physical means. These included mixing dry yeast cells, grains of quartz sand, and a soft, crumbly rock, rather like pumice, known as diatomite, and then grinding the mixture, including the yeast cells, with a pestle and mortar. The mixture became damp as the yeast cells ruptured and their contents were released. Then the damp mixture, which resembled a thick dough, could be squeezed to extract a pressed juice used in the experiments. This process was very efficient. Half a liter of liquid could be obtained from 1,000 grams of yeast, providing plenty of material to work with. When sugar solution was added to freshly pressed yeast juice, a strong production of gas occurred. In containers where the juice had been mixed with concentrated sugar, syrup, and left for several hours, there was active frothing of carbon dioxide bubbles, and a thick layer of foam formed, showing that fermentation was occurring. When sugar was dissolved in juice at blood heat, these effects were visible within 15 minutes. Careful investigation by Buchner and his colleagues established that carbon dioxide and alcohol were produced in exactly the same proportion as in fermentation with live yeast, but microscopic investigation revealed no living yeast cells in the extract. In further studies, Buchner found that the key chemical substance that promotes the breakdown of sugars in this way is an enzyme, which he called zymase. Zymase is a protein that is manufactured inside yeast cells, so in a sense, life is involved in a process of fermentation. As Buchner put it in his Nobel lecture, he was awarded the Chemistry Prize in 1907 for his biochemical researches and his discovery of cell-free fermentation. Quote, "The difference between enzymes and microorganisms is clearly revealed when the latter are represented as the producers of the former, which we must conceive as complicated but inanimate chemical substances." Unquote. But the crucial point is that the chemistry carries on whether the yeast was alive or dead. Enzymes are crucial players in many biological processes, but it is now possible to synthesize enzymes chemically without biology being involved. Okay, for the vocabulary section, we have much less than the story before. Respectable scientists, vitalistic process, refutation, refute, refutation, convert, conversion, physiological, physiological, inseparably, inseparable, separate, inseparate, separable, inseparable, satirically, satire, satirically, satirical, infusoria, intestine or intestine, urinary, encourage, encouraging, protoplasm, cell membrane, solvents, solvents, enzyme, enzyme, enzymas. Bom. Thank you very much for your time, and I will see you in the next story. Bye bye for now.